In this problem, we are given peptides produced by digestion of one and the same peptide with two different cleaving agents. And we know the primary structure of those fragments. And the first question that we need to answer is which cleavage agent is used for each of the methods. For that, we need to analyze the nature of the C-terminal amino acid residues of each of the fragments and need to deduce a pattern, because we know that cleaving agents, they cleave after certain type of amino acids. So here we see phenylalanine, tryptophan, tyrosine, and one which is odd, isoleucine. So most of those C-terminal amino acids are aromatic. So the agent that cleaves after aromatic amino acid residues is an enzyme chymotrypsin. So this is the agent used for method A. For method B, we have lysine, isoleucine, arginine, and lysine again. So most of those C-terminal amino acids are basic, positively charged amino acids. The cleavage is most likely done using another endopeptidase that cleaves after basic amino acid residues, which is called trypsin. So those two are our two methods. The second question is to determine what is the C-terminal amino acid residue of the original peptide, the one that we try to analyze, right? For which we try to deduce the primary structure. And remember, when we discussed, kind of tried to see what is common for those amino acid residues for each of the digests, we noticed that in both of them, isoleucine was kind of odd. And that's because when you cleave your polypeptide chain like that, you cleave after certain amino acids right there, right? But you cannot control and those certain amino acid residues would become your C-terminal amino acids of the cleavage fragments. But you cannot control the C-terminus of the very last fragment, because that's the C-terminal amino acid of the original peptide. And as you can see here, it's isoleucine with one letter code I. And finally, what is the primary structure of the initial peptide? In order for me to proceed, I need to locate the longest peptide that contains that C-terminal amino acid of the original peptide that we just figured out. And that would be peptide 4 produced by method B. So I'm going to write its sequence in the rightmost part of my paper, right? So this peptide encompasses peptide number 2 of method A, right? So this is peptide 2 from method A, but it also have additional 2 amino acids which I can locate in the C-terminus of peptide 3. So that allows me to extend my pentapeptide that I drew with arginine and glycine. Now I need to locate this arginine and glycine among the peptides of method B, and that allows me to write down tryptophan as my next one. But now I have a dilemma because I have 2 tryptophans right there. So I'm trying to see, where another tryptophan is located, and it's right there, right? So I kind of locate the whole thing right there in peptide 5 of method B. So it means that now I'm looking for peptide number 4 from method A, right? This tryptophan right here is that C-terminal tryptophan of peptide number 4 from method A. So I can extend this tryptophan with lysine, serine, and threonine. So I'm done with that one. Now, and I'm done with that one. So now this threonine, serine, and lysine are part of peptide number 5. So I can extend my sequence with the remaining amino acid residues. And my last one is tyrosine here. Tyrosine is present in the first peptide, so I can extend with lysine. Here I already used my 5th one, so the only lysine that I have left is part of my first peptide, method B, so I can extend it with phenylalanine. And the only peptide that I didn't use from the set produced by method A would be exactly the one that has phenylalanine as our C-terminal amino acid residue. So that allows me to extend it further. And just to make sure that I included all amino acid residues, I can actually count them in the peptide. So I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. So I'm supposed to have 21 amino acids here. And let me just count it. 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. So I accounted for every single amino acid residue. So that's my primary structure of the peptide that I analyzed.